Okay, thanks, Sven. Tremendously nice to be invited here to talk to you today. I work at the Receptor Biology Lab at the University of Antwerp, and here's my incredibly exciting title. It'll get more exciting, don't worry, hopefully. So, very thankful to Aubrey to bring up the issues of complexity in aging. We love complexity. Complexity is not a problem. It's a capacity to develop even more nuanced therapeutics. So, you just got to embrace it. So we embrace it by looking at cats. So, cell signaling complexity and hairballs. That's what we're really interested in. So, rough approximation, there are 1 million reactions every 10 seconds between 42 million proteins in one cell. That's the average degree of complexity we're dealing with. So, your cat having a good cough gives you this, a classical hairball. But what we do in the lab is we work on this, is we look at mapping interactions between different proteins in a single cell or even tissues, and it gives you another hairball. Can we find the order in this chaos? We're really interested in finding this because we want to then look for controllers of this chaos. So, we've already thought about how God interacts with biology in many ways, either from William Blake or from Einstein himself. So, the truth is that even in hypercomplex systems, there are beautiful orders. So, this is a beautiful picture of somebody taking a bicycle and pushing it 814 times across the room. And because the bicycle has a fixed structure, you develop this beautiful, I'll go back, sorry about that, this beautiful pattern forming just from something that you think is random. So, mathematics really does rule the universe, I'm afraid. So, I am trying to avoid maths because I got into biology to avoid mathematics. But a few years ago, I got really excited by these guys in their paper, Watson-Strogatz, looking at communication in complex network systems. And they did this beautiful paper looking at what they call small world networks, in which complex network connectivity was facilitated by these things in red, what they called loose connectors. And these have a very strong resonance with respect to functionality in biological networks for our lab. So, things like aging are involved in thousands and thousands of proteins. So, are they just randomly connected, or is there a functional hierarchy between them? So, does anyone read Tengway? Because this is the Lord of the Rings language, and this is the Dark Lord Sauron and the Ring. So, what we're interested in finding is there one protein to rule them all? At any one point in time, there will be, but it'll be a different one at different times. So, it's not a perfect rule, but it's a good place to start. So, what we think is that as far as cell signaling goes, there are small little microcomponents, and they form a modular network to control larger forms of functionality. But having network communication across this sort of thing is crazy unless you use these small loose connectors that Watson-Strogatz talked about. So we call these guys keystones, or we can call them hub proteins, but they're very specific. We think these guys are the future. They're very important to deal with therapeutic targeting of complex processes. So, we're interested in finding protein X, and I'll tell you about protein X today. So, this is what is a cool concept in biology at the moment, is looking at the fractal-like behavior of functionality in complex systems. So this is the beautiful natural fractal of a Romanesco cauliflower. It's very beautiful. So, if we're looking for a keystone protein, then let's look in a fractal nature. Let's look in the keystone tissue, perhaps. So there is a tissue in your body that is the functional keystone of many, many things. And it's this little guy here, the hypothalamus in your brain. So it is really the master controller. It controls so many functionalities. And so here's a nice MRI scan from Stanford, and it shows you that basically your hypothalamus projects to control cognition, behavior, endocrine functions, and autonomic nervous functions. And so it really does everything you ever want it to do. So we went looking for potentially the controller of the aging process here. So we did this in our poor unfortunate ratties, Sprague-Dawley's at NIH, and we looked across the whole age span from 3 to 24 months of age. We extracted the hypothalamus, and then we performed some protein analysis using these panorama cell signaling arrays, which allow you to measure 224 proteins in one go very quickly on a chip. And it's this differential dye labeling process. So pretty old tech, but it worked really well. So essentially, this is what we call a keystone analysis. So we're looking for proteins that actually could control the master controller and control as many processes in that master controller. So here's our workflow. So we found 85 significantly up or down regulated proteins in the old and and middle-aged ones versus the young animals. We did some very old-fashioned pathway analysis on these guys to get what's called KEGG pathways, and we found 12 of these guys. Then we did something more interesting. We wanted to look for, go beyond the classical form of pathway analysis and use a process called latent semantic indexing, which is basically a textual annotation process to look for semantic links across millions and millions of texts that no human can really possibly make. So what we were looking for here is we're using interrogated terms, which were the pathways, and we looked for all of the known protein associations that could be implicitly allied from biomedical texts. And what we did was create this great big matrix here of 0.3 million blocks of proteins that were related to the different types of pathway function. And there are some super proteins here at the top that are very strongly related to virtually all of these types of pathways, and these two guys in red here. So one was GRIT, and one's called GIT2. And here's these statistical analysis of this. And so these two guys are in the top 1 percentile of multidimensionally functionally associated proteins with all of these pathways in this master regulator organ. So, interestingly enough for us, GIT2 is a protein that we have seen interact with perhaps the most important therapeutic drug target in the world, and is the G protein-coupled receptor. This accounts for 30 to 40% of all known pharmacotherapeutics. And GIT2 exists in 3 different forms: GIT1, GIT2, and GIT2 short. And we saw the GIT2 isoform here, which is rather fascinating. So, this is basically an entirely theoretical prediction based on tissue analysis and an unbiased assessment. But what you can see here in the hypothalamus, when we tested the expression profile of GIT2 across this aging profile, you see this guy just keeping on trucking, increasing tremendously. The short isoform as well. The GIT1 does nothing. So this really is responding to the aging process. Or is it driving the aging process? We're not really sure. But we've seen across multiple species, like the rat, the macaque. Oh, the macaque's not coming up. Sorry about that. That is a macaque there. It's not coming up for some reason. It keeps on going up in the brain as you get older. We have then taken this to the next level, where we knock this out from animals. And you can see if you knock it out in males and females, you get a reduced lifespan and an alteration of their survival rate. It's a very complex process, this. I'm not particularly keen on doing lifespan extension studies in mammals because it doesn't mean a great deal. I'm more interested in the molecular pathologies underlying these. So, the interesting thing is, Morten has just shown very nicely that one of the things that regulates the way we measure aging is DNA damage. And GIT2 responds beautifully in a very similar way to what Morten was looking at these laser scratches in the nuclear DNA. So here's the gamma H2AX, which is the classical marker of DNA damage induced by the laser, and GIT2 goes right to it. And this is in 20 or 30 seconds, so it's remarkably quick. The nice thing is it also associates physically with this very important kinase here, which Morten actually showed in his talk as well, ataxia telangiectasia mutated, in response to ionizing radiation. And GIT2 is actually a substrate of this kinase and formed these beautiful foci in response to the DNA damage. Now, of interest, I actually work in the field of neuroscience, but you all know BRCA1 from breast cancer, but actually BRCA1 is also involved in dementia and many other types of neurological disorder. Cool thing is one of the functions of GIT2 is to actually stabilize the presence of BRCA1 in these DNA repair complexes. When you knock out GIT2, BRCA1 does not go to the DNA repair adducts in response to the laser burns of the nucleus, whereas p53 does. So it's fine. So it's a very specific function of GIT2. Now, when you look in these GIT2 knockout mice and look in their brain over a period of time, you can measure their sort of aging profile by looking at the gamma H2X foci in their cortex. And if you look at the difference between the young animals, since it's 4 months of age, the young wild types, and the young GIT2 knockouts, the GIT2 knockouts have a much higher stochastic rate of induction of DNA damage. If you look at older animals at 18 months of age, the actual GIT2 knockouts at 4 months of age have as much, if not more, damage than the 18-month-old wild types. And so these really are accumulating damage very quickly indeed. Now, the other interesting thing, as we saw actually from Morpen's talk as well, is, and I'm a huge believer of this, metabolism is everything. It's sugar, 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 sugar. That's it. That's what your body wants to do. It wants to do one thing. When you knock out GIT2, it screws it up. So mice have a beautiful pancreatic islet conformation in which the wild type, there's a beautiful beta cell core and an alpha cell halo around that expresses glucagon. What happens when you knock out GIT2 is the beta cell core has little holes in it and it gets filled up by these alpha cells. So what you classically do when you do an insulin tolerance test in these GIT2 knockout mice is you see they're insulin tolerant. They're insulin resistant. Classical sign of pre-diabetes. And this happens at a very young age, 2 to 6 months of age. So, not only does it affect DNA damage, not only does it seem to control the aging process in the hypothalamus, which is the controller of everything, it also seems to control energy metabolism. But this is just the first part of my talk. This is a new talk, so I haven't really shown it before. So, I think we should have a moratorium on lopazotine. I'm sorry, I'm never going to show it ever again. I've seen it 1 billion times now. We've got to stop it. So I'm not going to show you the hallmark of aging picture. One of the hallmarks of aging that I'm going to tell you about is the thymus, which I'm not an immunologist, and this is the first bit of immunology I've ever done, so please excuse any mistakes. So thymus is an interesting gland, and thymic involution, one of my friends told me, is one of the most important hallmarks of aging, so we thought, okay, let's transfer our work on GIT2 to look at some interesting work on thymic involution. So, these are the lymphoid tissues in your body. So there's the thymus gland, lymph nodes, spleen, and your bone marrow. Now the thymus gland is cool, so I learned this. It's always good to learn things every single day. That's why they're called T cells, because it comes from the thymus. So I learned that, so that's good. So hopefully you might learn that today as well. So it's the place for T cell maturation in the body, and it's really one of the controllers of the immune response. So as you get older, the amount of T cell DNA in your blood goes down dramatically, quite strongly. It's a 3 log 10 drop over lifespan. So it's really rather an important aspect of getting older is losing your immune function. So this is what happens to your actual thymic lobes. And so if you took a cross-sectional area, you'll see you get this fat infiltration and the loss of structure of the thymus itself. And this is called thymic involution, and basically it's a classical sign in virtually every single aging organism that we know of. So you're asking, so what is the importance of this? If you have an aging immune system, basically you have T cell decline, you have reduced resistance, which increases all of the types of disease, you have fat developing in your bone marrow, and also you have an increased incidence of tumor incidence with DNA damage, and of course an increased mortality. So, what happens with GIT2 knockout? And this is what happens. And so, this is the thymus of a 12-month-old, which is actually a relatively young animal. It's about 40 to 50 years old for humans. And this is the classical cortical and medullary structure, and it's largely lost in the GIT2 knockouts. If you look at an expression of a cortical marker called TROMA1, it's significantly reduced at this 12-month time period, which is still a relatively young and healthy animal. Now, the interesting thing is when you look in these GIT2 knockouts with respect to their thymic function, what you find is classical metabolic dysfunction issues in the thymus itself. So you see disruption of mitochondria, oxidative phosphorylation, NRF2, blah, blah, blah. All the classical things you associate with neurometabolic aging. So it looks as though it's a classical sign as well of what's going on with this GIT2 phenotype. Now, sorry for the grisly pictures. This is a dead mouse, and this is what a thymus looks like. So it's just above the heart. It's nice and white and quite large. The GIT2 knockouts are really rather unpleasant. So the thymus is sort of hidden away here somewhere. And then, in every single animal we saw at 12 months of age was this rather large lobe on the other side. We had never seen this before. This was called a parathymic lobe. That's what we called it, the PTL. So we asked ourselves, what the hell is this thing? Is it a lymph node? So this is transcriptomic data put in a PCA plot. These are the PTLs here, and these are the inguinal and the mesenteric lymph nodes from wild-type and GIT2 knockout mice. It doesn't seem to be a real lymph node, really. Is it a thymus? Not really, either. It's not really a lymph node. It's not really a thymus. This is wild-type thymus, and this is GIT2 knockout thymus. So this is a real funky thing, and this was 100% penetrance in every single animal. They developed this new organ completely at 12 months of age. So this GIT2 knockout PTL, the parathymic lobe, is a novel organ. So we looked in this thymus, and if you look at classical markers of thymic function, like sort of Lin-minor cells or early thymic progenitors or double negatives, you see the PTL is almost a unique entity, even different from the wild-type thymus. And the double negative 3s and the CD8 plus and the CD8 4 cells also seem to show quite distinct functionalities. So this really is a novel organ that we've seen. The interesting thing is, reports of this have been shown in humans as well in certain conditions. And we're now looking into those. But here's the cool thing. Nature finds a way. You know, like in Jurassic Park, you knock out the capacity to reproduce, they'll find a way to reproduce. The animal has found a way to get around the problem with its thymus. So if you do some transcriptomic analysis and you look at the differences between the parathymic lobe versus the really bad GIT2 knockout thymus, the GIT2 knockout thymus versus the wild-type thymus, and the PTL versus the wild-type thymus. I'm awfully sorry. It's very complex, but it's very simple to see. If you look at the proteins here in the middle, so this is a Venn diagram program that we've done that you can show different types of expression regulation in different sectors. And if you take these proteins that are common to all of these phenotypes, you can actually see that if you look in these PTLs, they are quite distinct from what's lost with the GIT2 wild-type, GIT2 knockout versus wild-type thymus. What that thymus loses is recovered in the PTL. So it's really quite remarkable, and it's all linked to do with aging and senescence. You can do this at pathway analysis level and see this even more clearly. So when you look at these 25 common pathways that are regulated between the really bad GIT2 thymus and the PTL in the GIT2 knockouts, what you lose in this badly disrupted GIT2 knockout thymus, you recover in completely the opposite direction with respect to pathway activation in this parathymic lobe. So all the functions it's lost, it's trying to reproduce in this other lobe. It's really trying to find a different way out. So it's really quite remarkable, the sort of perfect mirroring that we see. So this was very interesting, but we're interested in looking at GIT2's function across all lymphoid tissues, and so we looked to see what we call the multidimensional GIT2 signature. So if we looked across all the different types of lymphoid tissue we were looking at, the inguinal lymph node, the mesenteric lymph node, the spleen, and the thymus, this is transcriptomic data. You can see there's a core series of proteins here which are upregulated in all and downregulated in all. So we took these 40, and then we did some analysis based on a new program we created called Textrus. So Textrus is a natural language processing application which allows you to use small data sets and identify biomedically associated terms. So this is what Textrus looks like, and this is the URL. It's based at NIH where we created it. And so it uses this latent semantic indexing and a process called singular value decomposition to look for cryptic associations between terms and genes across virtually all biomedical texts. And it can do this tremendously quickly. So it can read 26 million pages, no problem, and give you results in less than a second. So the creation of this is used with PubMed abstracts, OMIM, and the Jackson Lab mammalian phenotype database. This allows us to actually end up with extracting noun phrases that specifically are related in a nth dimensional word space to your input series of query genes, which are called query vectors. And this is the point at where we simplify the term document matrices into these three different types of matrices here. So what you can create is you can create hierarchical word clouds, which look a little bit like ziggurats, essentially. So this is what a word cloud looks like. So the top of the cloud is the strongest associated words with your gene inputs. Specifically, this is called an agglomerative hierarchical clustering cloud. And obviously, the bigger the words, the higher the cosine similarity score, which identifies their sort of co-regulation in this nth dimensional space. And related words are put closer together. So it gives you sort of like a hierarchical clustering of the words related to your specific input. Also, in this program as well, you can create heat maps that show you individual strength of semantic associations between the specific words, and these can be expanded into noun phrases and the specific genes of your input. So we use this with this pan-dimensional GIT2 effects on lymphoid tissue in these animals. So if we take this data set here, where we show a common signature across all of these lymphoid tissues in response to the loss of GIT2, and we run this through Textrus, if you look at the heat maps, you get this clustering around clock regulation, transcription, response to metabolism of stress, and unsurprisingly, immune function. But this clock regulation is by far the strongest. When we try this with the other format, so the agglomerative hierarchical clustering process, with the same data set, you can see, you sort of get the feel it's got something to do with clock regulation in these animals. So what we did was we actually found that there's a specific regulation of a whole variety of clock-related genes across the whole lymphoid process of these animals. And that's really contributing potentially to the systemic aging process. So this sort of multidimensional controller of the aging process, GIT2, which we found completely by unbiased techniques, we think is an aging network controller. And we really think it has something to do with the time perception of aging, the control and integration of DNA damage with energy metabolism. Now, just published last year, this regulation of immune function, and also to specific stress resistance with respect to insults that affect DNA. So, GIT2 sort of does a lot. We've been working on it now for about 6 or 7 years. This is showing all the interacting proteins that are important in all of these different types of functional processes. And it does a lot that's linked to age-related disease. So this was just published this year, showing the role in these different types of disease that GIT2 has and the specific proteins that it utilizes to control these functionalities. So, just to finish, this is warping of space. This is called an Alcubierre warp drive signature. This is true. This is NASA's working on this stuff. This is how you do faster than light travel. And of course, this is Dorian Gray. So you can warp space, and you can warp the aging process in biology. So time, the progression of time, and the impact of time-related damage is entirely plastic. It's entirely dependent on how you regulate the accumulation of this damage. So, just to finish off, this is my lab at the University of Antwerp. So, I show you things that I don't really work on. I'm a receptor biologist. We work on creating drugs for diseases, essentially. This is work at NIH and at Duke and also MUSC, and these are all the people that gave me money to try and help people with disease. So thanks so much for your interest.
Regulation of clock has also been implicated in aging in other contexts. For example, the sirtuins, and does that play a role here, you think?
With the sirtuins, yes, definitely. So unpublished data, a lot of the SERT regulators also seem to control the expression of GIT2 responses. We've specifically looked at that in adipose tissue, actually, because it's been quite a nice place to look at, because one thing that I've ignored especially over the years, you're interested in, the metabolic control of aging. Sugar's important, but you know what? Fat's probably just as important, because the fat control is actually what keeps you being honest with the sugar end. So that's why we've been looking at the role of GIT2 and sort of PPAR gamma and also the SERTs in that thing. So it's not difficult to see how quickly you can hop between all the different major classical components of aging and how quickly GIT2 integrates them. And the reason it keeps on showing up these functionalities is we specifically went looking for something that would do that. So while it seems expeditious to say, oh, well, it does this and does that and does this and does that, that's because that's what we were looking for. We were looking for something that did that. And if we did this with other tissues and other types of organs, we would probably see the same thing, and that's what we've been doing. We've been done like an age map, essentially, of these GIT2 knockout animals, and you keep seeing this signature everywhere. But good question.
And one last quick question, maybe? So it seems like you've shown that it's like a really central regulator in a lot of these age-related processes. Yep. If you knock it out, that's bad.
That's the question everyone asks me. Is it good? Is it bad? It depends at the certain time.
Okay.
To me, this is the thing I discuss with my students all the time. Is a sign of stress a good thing? You could take it from the point of view of, if you show signs of stress, it means that you didn't respond to the stressor causing the stress, and there's an extant demonstration of an insult. So people always ask me, is it good to knock it out, or is it good to overexpress it? I think it's a horses for courses scenario. I think at certain times it might be good. In general, the overall theory that I would say is GIT2 is an integrator of the sugar metabolic process. It's there to control and regulate and it's a far more complex process than we ever thought. It's got something to do with fat tissue, it's got something to do with glycemic tissues. It's a systemic-wide process. So in different areas, you might see it actually has different functionalities depending on what you really want. So what we're trying to do, because I'm a receptor person, we're looking for the receptors that interact with this thing. And we've identified some very good ones, and we've already shown that actually, here's the news. So for a long time, GPCRs have been considered to be just G protein signaling controllers through G proteins. Then 20 years ago, I specifically worked on a project that showed it to be controlled by beta-arrestins. And so we went from one dimension of signaling to 2 dimensions. GIT2 is the 3rd dimension of signaling. It's not a regular signaling process, but it's a signaling process in the context of stress. So all of these other different types of cell signaling don't readily show themselves in most experiments, but when you do a perturbation, these things pop up. These receptors then choose different types of process to go through. That's what we're trying to identify. What are the specific conformations of certain types of receptors that actually go through this protein as a signaling protein? That we consider them as being like sort of like an aeroplane with a parachute with GIT2. They hold it at certain times and they drop it at certain times. And when it gets dropped, it does this functionality. So it's a good question.
Thanks. So beyond the molecular biology, do you think it's potentially a useful target for pharmacological interventions or not?
Oh, absolutely. We're already there, don't worry. We already have molecular variants of proteins that do regulate the signaling through this protein. So, oh yeah, we're always looking for VC, don't worry. It's
okay.
Okay, thanks.